Hello, everyone. Thank you for joining our webinar, It's the Little Things That Count, Quantifying and Characterizing Microplastics in Bottled Drinking and Source Waters. This webinar will be hosted by Dr. Mike Dziewatkoski, Technical Director of Eurofins SF Analytical. I'm Genevieve Randall, and I'll be moderating this webinar. Before we begin, I'll let you know more about how this webinar will run. The webinar is being recorded, and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer viewer-submitted questions. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions throughout the webinar. Now I'll give a quick overview of Eurofins. Eurofins is driven by our mission to contribute to global health by offering the highest quality testing, training, auditing, and consulting services. We strive to listen to our customers and not simply meet but exceed their expectations. Our footprint is global with 45,000 staff in over 800 laboratories across 47 countries and a portfolio of over 200,000 analytical methods. Eurofins provides a unique range of analytical testing services to the pharmaceutical, food, environmental, and consumer products industries and to governments. And Mike, you may start your presentation.
All right, thank you, Genevieve. Good morning, everyone, or good afternoon, everyone, wherever you may be. And thank you for taking the time today to join us for this webinar. Today, I'm going to be discussing the work we do in testing for microplastics in water. Now, although I'm going to be focusing primarily on water, this testing can be applied to a lot of different food matrices. So any type of food that's harvested from a water source or even from a terrestrial source. But primarily today, what we're going to be talking about are why microplastics are such a concern. So why is there so much press? Why are we hearing so much about it? So what is this all about? I'm going to be talking also about why we're doing the testing. So who needs the testing, who uses the results, how the results are used. And I'll be talking about a little bit about the different techniques we use to make these measurements. And then I'll be talking about how samples are collected and managed to prevent any type of outside contamination, and then the actual methodology behind the testing. So specific test methods that we employ or even hybridized methods. And then we'll be discussing about the typical results that we obtain, and then how those results are used by our clients. And then finally, I'll be discussing a little bit about the future of testing. Right now, it is really important to realize that there are no standardized test methods for determining microplastics in any matrix, whether it's water or solid food. And what we're doing right now is we're working towards the day when we will have standardized methods that are approved. And that's one thing I'll be spending quite a bit of time on it and discussing where the future will lead. So let's talk a little bit about the background of microplastics and why this all became such a concern. Well, unfortunately, there's bad news and good news. So first, the bad news. Plastic contamination is everywhere. Plastic litter is really prevalent anywhere you look, whether it's in water, on land, and even you can find small particles of plastic in the atmosphere. The reason being is for well over 100 years, we've been using plastics in some form. Starting about 50 years ago, a lot of products that were made with metals or maybe glass were replaced with plastics. And so this plastic, unfortunately, has wound up in a variety of different areas. So, for example, plastic that is just discarded, like, for example, a plastic boat dumped into a river. Plastics that are degraded and introduced into the atmosphere as tiny particulates. There were once plastics, little microbeads, put into personal care products, like body scrubs and toothpaste. And then we also find plastics coming from a source of runoff. So anything that's deposited on land is running into the water sources. Then finally, we produce plastics every time we do laundry. Our clothes are made out of polymer fibers, and those fibers break down during the washing process, and that's introduced into water. So you have many different ways in which plastic waste can enter into water. And this can be a freshwater source or saltwater source, so pretty much any water source you can think of. The types that are present are multiple, and these are just a few listed here. And one way or another, you have more than likely encountered these different polymers in your everyday life. But since we're using these in our products, we're discarding these in waste, we're wearing these different polymers, you're going to find these end up in a water source. And so that's where the concern lies, is that we are experiencing water contamination as a result of plastic. And also now since we harvest a lot of our food from water, we're seeing that contamination from water into our food sources. So let's take a closer look at different plastic sizes that we encounter. You know, plastics come in all different shapes and sizes, from large particles, stuff that's greater than 5 millimeters, all the way down to what we call a nanoplastic. And these are less than, considered usually less than 1/10 of a micron, so they're tiny. But primarily, today's talk is going to be focused on the microplastics area, where a lot of emphasis has been placed recently. So to give you an idea of scale that we're looking at sizes that are about 1 micron up to about 1 millimeter. Now, this isn't a hard and fast rule in terms of a category of sizes you look at. We can expand this as we want. So, for example, if someone were to come to us for testing and they wanted to look at particles that were maybe up to 2 millimeters, we could do that, or 5 millimeters, we can do that. So the test methods can be modified. But to give you an idea of scale, on the upper end of the range, salt crystals, ordinary table salt, is about 300 microns in diameter. So that's easy to see with the unaided eye. So you can pour some out on the table and you'll see where you've spilled the salt. On the other hand, at the lower end of the range of size, we can look at red blood cells. And red blood cells you cannot see without some sort of microscope, so you need some magnification. And those are on the order of 10 microns, maybe sometimes a little bit smaller. So we're talking about things that you cannot see with the unaided eye, up to very relatively easy to see pieces of plastic. Now, when we're considering what we find in the environment, we can characterize plastics in terms of their morphologies, so their shapes of the plastics. And any type of plastic shape you can think of, we can find. So for example, we find fibers materials, and these fibers can be single strands that are appearing in a water sample. We find fiber bundles, so these are multiple strands of fibers wound together. We find sphere-like structures. These could sometimes they're almost perfect spheres, and then sometimes slightly irregularly shaped spheres of all different sizes. We also find plastic film. So if you think about a film that you use as plastic wrap in the kitchen, only now think about it maybe finely chopped up, you can find actually little bits and pieces of plastic film in some water sources. And then there are pieces that we consider as fragments of bigger parts. So bigger pieces of plastic that have broken down, degraded for some reason. Maybe it's action of sunlight or rain. So the elements in some way break down bigger pieces of plastics into tinier fragments. And then we have foam-like elements. Think about polystyrene or polyurethane. Polyurethane in padding, and then polystyrene that's used in coolers. So it's that type of a foam-like structure that we'll observe. So now we know that plastics are quite prevalent in the environment, and they can be present in very tiny sizes. And so what we have to start doing is looking at what impact this has on food sources and water sources. And if you think about it now, if you have microplastics in water where aquatic life is living, then you can, and we know this happens, we know that plankton, like zooplankton, will actually ingest microplastics. And so you have now an animal here that has ingested microplastics. It is eaten by a larger animal, like for instance, a small fish. Then the small fish is eaten by a larger fish, and eventually it works its way into us because we eat the large fish. But what's also true is that since these microplastics can be quite small, small fish can eat plastics, and even the larger fish can eat the plastics directly without first eating another food source. And just to really illustrate just how prevalent this is, and also some really neat work being done by researchers, I put a link here to the National Public Radio site. And if you follow this link, towards the middle of that web page, you'll see a really neat video where they're talking about just the contamination of shellfish with microplastics and what they're finding. It's a really great illustration of what's happening. And so practically any food we harvest from water can contain a microplastic. And here are some examples. Microplastics have been found in canned sardines, in shellfish, in seaweed that's used to make sushi, and even in sea salt. So, you're taking seawater, evaporating the water off with sea salt being left behind. Well, there's a potential there for plastic particulate material being present. So, again, many different routes here. And so this multiple routes of exposure really raises quite a lot of concern in terms of what's happening from a biological fate standpoint. What is happening to these once these microplastics are being ingested? So there's a lot of emphasis now on studying what's happening in the food chain. Are these causing disease and so on? What's happening with respect to the presence of all the polymer additives that are in the microplastics? So almost all plastics have some type of additive. So you have plasticizers and extrusion aids and so on. So different additives to the plastic that modify its characteristics. And now when a microplastic is embedded in tissue, what's happening with those polymer additives? Now, since microplastics are hydrophobic in nature, they're lipophilic, they will absorb organic compounds quite readily. There is a lot of interest in what's happening with respect to adsorbed and absorbed contaminants. These could be PCBs, pesticides, herbicides, anything that's out there in the environment, and we know those are out there in the environment, then the microplastics could be actually really concentrating that contaminant and then transferring it to any type of element, any type of living organism that eats that plastic. Well, now that we're done with the background, let's take a look at some of the testing and really answering the questions of why we do the testing, why is it important, and how is it done. The reason this all became a lot of concern was early work in environmental research, really studying what is happening in sea life with respect to plastics contamination. It was found that there were many different forms of aquatic life that were ingesting plastic in the environment. Not to mention the fact that you can go to almost any beach now and find plastic waste. You can go out to the middle of certain oceans like the Pacific, and you'll find different pools of huge reefs of plastic. Unfortunately it's quite pervasive. Now, in terms of regulation, well, I'll speak to just what's happening in the United States. Because of contamination from microbeads, there was a Microbead-Free Water Act was passed in 2015. So eliminating the use of microbeads in personal care products. And that was a good start. But then now, more recently, California has taken the lead on really introducing legislation, passing legislation that requires that the state really address microplastic contamination, really have a strategy, a plan in place to deal with it. And part of that, as a start, was dealing with it in terms of what's happening to drinking water sources. And that's not a source water, but also finished drinking water. So specifically, SB 1422 is going to require that we have a formal definition of what a microplastic is. I gave you a definition in terms of size. That's going to be more formalized in this regulation. And then going to have to actually adopt standardized methods. Now recall I mentioned that there are no standard methods at this point. We know our methods work, but really what we need to do is get many other groups involved, some regulatory institutions involved, to really hash out the details and to prove out exactly how we're going to perform this testing. And then there's a requirement that there's going to be required 4 years of both testing and reporting results for water sources. Now, in addition to environmental and regulatory, there's also just product quality assurance and quality control. And there's been a lot of interest from bottled water producers in terms of studying, well, number one, answering the question, is our product contaminated with microplastics? And if it is, then could we figure out what is the source? You know, if we know what the source is, maybe we'll be able to correct it. And then maybe even leading to us changing the way we package our products. So improvements in packaging processes, packaging materials, these are all important to being able to maybe improve the level of contamination. And then there's also the public relations part of it. So consumers want to know that what they're drinking is safe and wholesome. And so that's an important part to this, is ensuring the public that, yes, indeed, what they're ingesting is safe for them. Now, recall I mentioned there was early on, I mentioned those bad news and good news. Well, the good news is we have good ways of detecting microplastics in both water and also some food matrices. And so we use techniques that are, well, some are simple and some are very complex. We can start with just something simple like an ordinary light microscope that you'd find in most microbiological labs. And you could have magnifications up, sometimes up to 400. And simply just using ordinary light microscopy, you can see microbeads, you can see microplastics. And so with this, we could do actual measurements of size. We can look at their shapes. These happen to be pretty consistent spheres. And then we can also maybe even look at, well, characterize their color. So are they opaque? Are they blue? Are they red? Are they green? Because sometimes a color tells us a lot about what the source is. Now, if we want to get more elaborate, we want to be able to determine what is the chemical identity of that plastic. So using ordinary microscopy, I can't do that. But what if I could isolate these beads, in this case, and do some spectroscopy? And indeed, I can. I can use what's called a Fourier transform infrared spectrophotometer to do a measurement of and determine chemical identity. So literally, I could pick out a bead, if it's large enough, I must say, and then transfer it to an ordinary benchtop system and get a spectrum. Now, if I really want to get fancy, I can automate this. And so what I could use is what's called a micro FTIR or even a micro Raman system. So it's a microscope with a spectrophotometer that's going to allow you to collect information about a lot of different particles and automatically go through and pick out particles and get chemical information about those. Now, there's another technique that's often used, and that technique is based on gas chromatography. But the interface to it is what's called pyrolysis. So what you literally do is you take the plastic material, you heat it up to where it starts to decompose, and you send fragments of that chemical plastic, the chemical compounds, into a gas chromatograph that is equipped with a mass spectrometer. So what you get, if you recall, if you've ever done any work in gas chromatography or in liquid chromatography, recall that you get a graph of response as a function of time. And you'll see different peaks appear for different chemical compounds that are present. So this technique gives you retention time and also some mass data which you can use to actually identify chemical compounds. Now, before we continue on any further, what I want to do is get some feedback from you. And so we're going to ask you to take a polling question.
All right, thank you. And I'm going to launch the first polling question now. It should be on your screen. And that question is, do you expect microplastics drinking water regulations or consumer concerns about microplastics in food to impact your business? Answers are yes, will be impacted by drinking water regulations. Yes, we'll need to address consumer concerns related to food. No, my organization will not be impacted, or I am not sure at this time. I'll leave the poll open for about 30 seconds before I close it and share the anonymous results. Okay, I'm going to close the poll now. And it looks like the most popular answer was companies being impacted by drinking water regulations. And I'm going to show the results. Okay, and Mike, you can continue with your presentation. Thank you.
All right, thank you. And thank you everyone for participating in that polling question. Let's move on to talking about actually some of the testing we do. And this will be primarily starting off initially with how we collect samples, work with samples, and then the actual details of some of the methods. Now, I want to point out that although we're focusing primarily on drinking water here, these methods can be used with some adaptation to the analysis of solid food matrices. So let's take a look at the different types of samples that we test when it comes to water type samples. So we could have samples coming from salt water, fresh water, drinking water, so fresh water that has been treated in some way and sent through the drinking water distribution system. We can look at bottled water, and sometimes bottled water sometimes it's highly purified, and then sometimes it comes just from a tap. And then also we can look at gray water. So gray water is water for reuse. This is stuff that comes from the laundry, for example, maybe from the bathtub or the shower. It's what's considered gently used, and in places in the country and across the world actually where water is very scarce, this is becoming a very important source of water for use and conversion into drinking water. But as I mentioned earlier, that when we do laundry, we are producing microplastics and they're getting into the water. So gray water, you could expect potentially to have microplastics to be present. Now, how we sample water is depending upon how the data that we're generating is going to be used. So, for example, sometimes if it's a surface water, or what we call environmental water, you might literally pull a net through the water and collect particulate pieces of plastic. And these tend to be somewhat larger, greater than about 0.3 of a millimeter. Or you could literally dip something down into the water and grab a sample. That might be 1 liter, 2 liters, maybe 10 liters. Now, if you're talking about drinking water, usually you're collecting a sample somewhere in the distribution system. So that might be at the final tap, or it could be right after primary filtration, right after secondary filtration, so somewhere along the line. And then bottled water, that sample is analyzed exactly as it is bottled. So it's shipped to our lab, and we open it under a controlled environment and test it. Now, when we're collecting samples, we have to make sure that they're dealt with properly. So we have to have a formal chain of custody. We have to know who collected the sample, when it was collected, where it was collected from. Because what we want to do is make sure that that sample is in no way altered before it enters our laboratory. Because in a lab, we have a very clean environment. So what we do is we make sure that we minimize plastic contamination. That's thorough cleaning. That's we're not working with glassware, working with glassware and not working with plasticware. So we're trying to avoid plastics as much as possible. We work with very high purity water. And we will test our water. We will test it to make sure that it's free of plastic particulate. And then we use what are called blanks, and these are literally essentially samples of water left open in our laboratory to see what the impact of just background contamination is. Now, this work is done in what's called a laminar flow hood. So these are hoods that are designed to prevent any air from the outside of the laboratory entering into it. So it's an enclosed environment, and the air that enters into the hood is filtered. So it's very carefully controlled. But what we do here is this is where we do all of our sample manipulation, all of our prep work. Our reagents are stored there, and this is where we do our microscopy. So all of this has to be very carefully controlled. Now, in terms of the actual methods we use, we primarily rely upon two different ways of doing the testing. One we refer to as a dye imaging method, and this came out of initially from the State University of New York at Fredonia. And this is primarily used with clean water sources. So this could be drinking water or bottled water. So there's almost no particulate organic matter present that wouldn't be a microplastic. It's going to be primarily highly purified, highly filtered water. So relatively clean stuff. There's a second method that we refer to as the NOAA method, and this is designed for relatively dirty samples. And this is a picture of a typical sample that you would see with a lot of leaf matter, a lot of organic matter, plant material, leaf stems, twigs, etc. Maybe some dead animals from the water source, so little water bugs, and that all has to be removed. But the analysis workflow, regardless of which method you're using, goes like this. We want to collect the sample, and and how we collect that sample depends upon the type of study we're doing. So is it going to be an environmental water, or is it going to be drinking water? So what is the purpose of the study? Most importantly, though, that sample has to be representative. So the question becomes, do you collect just 1 liter of water, 2 liters, 10 liters, or even more? Well, in some respect, those questions aren't answered, and they are dependent upon the type of information you're trying to generate. We then, once we have the sample, we go through a step of what's called isolation. One way or another, we're going to treat that sample. We're going to chemically treat it to modify it so that we can do a separation of particles, the plastic, from all the other stuff that's there, or maybe some type of filtration. So we're going to somehow isolate the microplastic particulate. Then once we have that plastic, we're going to do measurements. And we can collect a lot of data, actually, in doing the measurement process. So for example, we can count the number of particles we find, or if there are enough, and just the quantity is large enough, we can get an accurate mass. We can also characterize, using microscopy, the shape of particles, and even so the sizes and colors. And then we'll do an identification step. And this isn't always done, but sometimes our clients want us to tell them what is the chemical identity of the plastic that you have found. So let's take a closer look now at the dye imaging method. In this case, what we're doing is we're working with the bulk water sample. So someone has collected a sample in a bottle. In this case, it's a plastic bottled water. Sometimes it's collected. Samples are collected in glass jars, sometimes metal jars, and then that's sent to our laboratory for testing. What we do is, working on the entire bulk sample, we'll add a special dye called Nile red dye, shown here. This dye is very lipophilic, and it tends to adsorb and absorb into plastics quite readily. And it's pretty good, actually, at being absorbed to plastics, preferentially over other types of materials. So for example, inorganic materials like grains of sand won't be dyed by this chemical. And to a large extent, other matter you'll find, like pieces of water bugs or crustacean shells, also won't be dyed. It's not perfect, but it does a pretty good job. We then let the sample incubate for a little while with that dye, make sure it absorbs really well onto the particles. Then we'll do a filtration step. And here we're using a 1.5 micron filter, so we're capturing everything larger than that. But there's no reason you couldn't use a larger pore size filter or a smaller pore size filter, depending upon what the nature of your study is. And then we expose what is captured on the filter to short wavelength light, somewhere in a range of 500, 510 nanometers. And if you look at the fluorescence that appears at 530 nanometers or greater wavelength, you'll see little glowing dots on the filter. And so those are relatively easy to see compared to undyed plastics. And so it makes it possible to go in and actually count them and characterize these things. So we go through a step where we're going to actually do some microscopy to come up with particle counts and also determine sizes. So 2 key pieces of information. Now, in contrast, the NOAA method is for dirtier waters, and the sampling sometimes is performed differently with a net. And we're typically looking at larger particles. Now, a key part to this method is that there is a chemical oxidation step to remove as much of this organic matter as possible. And this works quite well, actually, in removing practically all of the organic matter and leaving the microplastics unaltered. So this type of method is very useful for something that is dirtier in nature. Then what's called that the solution that results is it goes through a density adjustment so that you can make the plastic particles float. You can collect those, and then once you have the plastic particles collected, you can perform microscopy and then go forward with either a count on the number of particles you found or determining mass. So the two methods are related in that in the end you're isolating plastic particles that you're going to characterize, but they focus on slightly different matrices. Okay, now we'd like to have your say in this a little bit more, and so we'll take another polling question.
All right, and the new question should be up on your screen now. That question is, which statement most closely describes your interest in microplastics testing? And answers are, we currently have our products tested by an outside lab. We currently do testing on our products in-house. We plan to start testing in the near future, or we will probably not have a need for this testing. Select your response from the polling question answer choices, and I'll leave the poll open for about 30 seconds. Okay, I'm going to close the poll now and show the anonymous results. And it looks like a lot of you plan to start this type of testing in the near future. Okay, and Mike, you can continue with your presentation.
All right, thank you, Genevieve. All right, now let's get down to the best part of the presentation, and that is with respect to what kind of results do we see, and how do we use the data, and then where are we going with this in the future? Well, the typical data we see is that we will come up with, for the volume of water we've analyzed, a number of particles, usually typically per liter. So we might usually we test either a liter or 2 liters of water, sometimes more, but we'll come up with a count per liter. Or we may come up with the mass of particles determined per liter. In addition, though, we will provide information in terms of particles found into 2 different size groups. Now, in this case, we typically pick stuff that's less than or equal to 100 micron and then greater to 100 micron. But these two categories are arbitrary because we can adjust those as needed. So that's pretty straightforward to do. And again, there's no standardized way of reporting the data. It just depends upon the study that's being performed, the requirements of the data use. In addition, we'll come up with the shapes and of the materials found, and also the colors that are found. Again, color sometimes is a very important piece of information. And if requested, we'll perform spectroscopy to determine chemical identification. So if we're doing this work manually, we typically have to work with particles that are much greater than 100 microns because we're manually pulling off these bigger pieces of plastic and transferring them to a benchtop FTIR to collect some spectral data. So, for example, from our sample, we may see a spectrum like this. And so this is just a plot of the amount of light transmitted through the sample as a function of wavelength in terms of wave number. And where these negative going peaks appear tell us something about the chemical nature of that sample we have tested. So that's important information. What is really nice, thanks to the modern era of fast search computers, we can take this data from the actual sample and have it compared to multiple spectra in our library of spectra that we have and come up with actual matches. And it just so turns out that although we have a huge library, if we need to add new materials to the library, we can always do that, because you can always obtain a reference material and do a spectral scan on that and save it to your library. Now, sometimes we don't want to do this manually because we want to actually have this work done all by itself, maybe even a little bit faster, so no human or little human intervention. And so we move to micro FTIR, in which case we're taking all of the particles that are captured onto a filter bed, and we're doing a couple different things. Well, we can enhance the image, change the image to give us what would be a high likelihood of detection of a microplastic, and focus on the different particles and do a couple different things. We can do microscopy, so we can get a magnified image, determine sizes, and then for each particle, we can do a spectral scan. And then these systems also have libraries of spectra, so we can do comparisons to our library data and then come up with a match. And so in the end, when we're providing someone with data, we provide them with actually quite a bit of information. We come up with particle counts and morphologies. So literally we can show them images of what we found, spheres or fragments, fibers like this, branched fibers, or even linear fibers as such. Now, typically what we do is we provide information from about for particles from about 5 micron up to about 2 millimeters. The upper end is pretty easy to change, so we can modify that as needed. Now, the lower end of this scale is dictated by, first off, how accurately can you measure the size, and then how accurately can you, or how reliably can you collect spectral data. So size-wise, it's not too difficult to make measurements at the 5 micron size with an ordinary microscope, but with an FTIR system, it is very difficult to get chemical information at this small size. In addition to this size range and giving an imagery of what we found, we can also report on what shapes we found. And then if we're able to isolate to larger particles, we can do some spectral identification. So we give a chemical identification, determine and report, was the material polyethylene, polystyrene, polypropylene, or some other type of polymer. Now, whether this data is used by someone for drinking water or bottled water, you can use that information to maybe come up with a way of determining what is the source of that contamination. So for example, is the source from packaging? Is the source from a piece of bottling equipment? Is the source from the distribution system of the drinking water? So those are all good questions, and those are all potential sources of contamination. So this could lead to improvements in filtration, bottling, and packaging. Now, in general, we report data in this format. So this is an actual snippet of a report. So what you'll see in a typical report are size ranges into 2 different categories, and with a number of particles reported in each category. And note here that in this case, we tested a 2-liter volume of water to generate as one sample. In addition, we will, for certain particles, it depends on how many a client wants to characterize, we will determine what is the material. So what is the chemical identity, in this case polypropylene, and happen to be a fiber. And then we found, in this case, some polyethylene, and it happened to be a fragment. Now again, this data you can change in terms of your categories of reporting and also the number of particles you actually want to report. So this data can be reported as needed. Now what is true about this is that there is no standardized way of reporting data at this point. But in the future, what could you expect to see? Could you expect to see that you're still going to have to report some sort of quantity, the number or mass? Size is pretty important, very important actually. Maybe even the shapes, because it can lead to identifying potential source, as can chemical identity. And then something also that's going to have to be reported, and there's a lot of discussion about how to do this right now, and that is, what is the detection limit, or a limit of quantitation? So what is the tiniest mass I can measure for a particle if I'm using Pyrologist's GC mass spec? Or what is the smallest number I can count in this particular size range? So those are all good questions, and those still have to be answered how we're going to formally report that data. So in the future, we can ask the question, are there better ways of doing this testing? Are there better methods we can rely on? And EPA also had that question. So in 2017, they held a workshop and collected a bunch of experts in the field of microplastics. And although this workshop covered many different areas from contamination all the way to new test methodologies, one thing that came out in the report was of great importance, and that is that they realized that standardized test methods do need to be developed. And more than likely, this is going to happen by including multiple different organizations, professional organizations, industry, academia, research institutions, so that based on what we have in the research literature right now, the methodology that's used, we can take that and do some further development, further refinement of those methods, and then ensure that they work. And the way we're going to ensure they work is to have multiple laboratories involved in applying these test methods and verifying that they work and looking at the quality of the data. In addition, there's a lot of interest in quality assurance and quality control. And primarily that leads back to being able to account for background contamination. Now, I started off in this presentation mentioning that plastics were everywhere. So you have contamination everywhere. So we need to have formal practices in place to be able to evaluate background contamination using blanks, being able to spike in known amounts of plastics so that we can be able to determine the recoveries and make sure that, are we getting accurate counts? Or what is the accuracy of our recovery? Okay, and what is the precision, analyst to analyst precision? Now, in order to do that, we're going to need some very well characterized standards. We rely upon some what we call reference materials, and they're not considered certified reference materials, but they are reference materials for plastic particulates that we work with. But in the future, we'll probably see this expanded to a lot of different materials. And then some statements about a limit of detection, limit of quantitation, and also guidance regarding how samples are to be handled and how preparation is supposed to be carried out. So all very important stages to ensuring quality. So in summary, we talked a lot about plastics and how the fact that plastic waste is everywhere. It's unfortunate. It's something we have to deal with. It's not going away, but it is a huge concern, primarily because plastics, we know for sure that plastics are entering the food chain. There is a lot of concern about toxicity. We know now that current and then future regulations are going to drive a lot of the testing that's being performed. Fortunately, we have many good ways of characterizing plastics. We can do that with microscopy, spectroscopy, and chromatography. And in one or more ways, multiple techniques will be used. And then eventually, we're going to come to a point where we're going to have standardized test methods. We're not there yet. The methods we do use, we know they work, but it's going to be good to have everybody on the same page and to ensure that we're generating useful data. I'll say, okay, so with that, I'll thank you for your attention, and I will hand it back over to Genevieve.
All right, thank you. Okay, so now we will move into a short Q&A session. Feel free to continue to submit questions during this session. We'll follow up with any questions that we don't get to. And our first question, what is the smallest particle size you can characterize?
Yeah, that's a good question, and it does depend upon the technique that you're using. So right now, when we're talking about characterizing a particle in terms of chemical identity, for the most part, if we're using FTIR, a micro FTIR system, we're limited to about 20 microns in size. Now, if someone is using micro Raman, they can move to much smaller sizes. But there are advantages and disadvantages to both techniques in terms of the plastics that are amenable to being tested, size, and also speed. So there's a little bit of variation there. In terms of, if you're using ordinary microscopy, you can, size-wise, you can determine morphology down to maybe 5 or fewer lower microns, so they're real, very small particle sizes. You could even use scanning electron microscopy to generate data in the nanoparticle range. So yeah, it just depends upon the technique being used.
All right, thank you. Our next question, are the test methods applicable in food?
Yes, they are. And what we do is we typically use a hybrid method. So for example, we may take the dye imaging method, parts of it, and combine with the NOAA method. So what we'll use is, for example, in some food matrices, we will oxidize all of the material present using chemical oxidation, peroxide oxidation. And then material that is left over, the plastic particulate, will actually dye. So we'll hit it with Nile red dye and stain it, and then collect fluorescence information from that. So we can combine methods.
All right, that actually ties really well into another viewer question. Will the Nile red dye non-plastic organic materials?
It can. It's pretty good at specifically dyeing something that's very, very hydrophobic, like a plastic material. But there is plenty of work in the research literature showing that it can sometimes dye other organic material. So you can't rely on 100%. And we will see that sometimes we'll see something dyed that may not actually be a plastic, but we can usually figure out if it is by other techniques. If it's large enough, we can do chemical identities. But sometimes it will dye non-plastic materials.
All right, thank you. Next question is, and I want to remind everyone watching still that you can continue to submit questions throughout this Q&A session. The question is, in sampling, using a netting apparatus approach, is the length of time for sample collection considered a factor? That is, if the collection were over a 24-hour period, for example.
Yes, so that information is collected. So it's known, for example, how far the trottle net was pulled, and then for length of time. So yes, all that is important. And it's surprising, some of the information I've seen come out of that sampling. That net can fill pretty quickly. Not completely full, but you can acquire relatively quickly a sizable amount of plastic. So yeah, the time is a factor.
All right. Okay. Okay, I'll ask you another question. What is the suitable volume of water for bottled water in order to generate accurate results?
Yeah, so that's something we typically will look at. Sometimes we look at individual bottles. So if it's like a 500 ml bottle, we'll look at an individual bottle. And the reason that's important is that you limit it to the amount of that's actually been bottled, is that sometimes there's a packaging issue that needs to be worked out. So sometimes it's a 500 ml bottle of water, sometimes it's a liter, a 3/4 liter. So that can vary as well, but the important thing is that we do know exactly the amount of sample we have, and then we express the results appropriately. But it's and then the question becomes, if I test, let's say 20 500 ml bottles of water, and I'm seeing particle dispersions that could be one bottle has 20, another bottle has 500. There's something going on. There's an issue there. Is it contamination in the package, or is it in a bottling line someplace? So variability is a problem that has to be looked at.
Okay, last question for the Q&A session. Collecting grab samples in drinking and wastewater treatment plants may not be representative enough. Do you have a recommendation on sampling for these sites?
Now that's a great question, because the grab sample method is being discussed in terms of being potentially more representative, and the reason being is that people are also collecting samples using pumping systems. And the pumping systems, you can acquire a large volume of water pretty quickly, but the problem is there's associated with that pumping system plastic material, and actually the transfer lines, if they're a soft plastic, then they're contributing to contamination, and that's been observed. So sampling in a drinking water system is still going to be a challenge. But yeah, so I would not, if you're doing it on a pumping system, you have to look at background to see what's coming off the pump.
Okay, well, that wraps up our Q&A session. Remember, any questions that weren't answered during the session will be followed up with on an individual basis. And I want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available to you within 3 business days. This will come to you via email. Thank you, Dr. Mike Dziewatkoski, for your expertise and presentation, and thank you, everyone, for attending.
Thank you.